housekeeping rules that mute your microphone during the talk. And if you have any questions, you just put them into the chat box, and Rajesh will answer them after his talk. So just to give a brief introduction, Rajesh did his PhD at Heidelberg University in Germany with Matthias Seedorf on the topic of STIM and PIP2 interaction. In 2014, he moved to Switzerland, and he's working as a postdoc and research associate with Matthias Hediger. And his main focus of research is actually STIM-ORA activation mechanisms, inactivation by calmodulin, redox modulation, or ORA modulators, and so on. And today, Rajesh will give his talk about the activation mechanism of ORA channel isoforms. So we're looking forward to your talk.
So welcome, everybody. Thank you for the introduction, Irene, and thank you, Martin and Katja and the ECS to allow me to present my work today. So as Irene mentioned, I'm going to talk about the activation mechanisms of ORI channel isoforms, basically using ORI mutants that are thought to mimic the STIM1 mediated gating. And this morning I was building this house with my son, and I thought that this could be nice gates, ORI1, ORI2, and ORI3. And since we know more about the activation mechanisms of ORI1, so you see that the gate is open, but for ORI2 and ORI3, not much is known. So what I will do is give you a brief introduction about store-operated calcimentry. So under resting conditions inside the cell, you have the ORI channels in the plasma membrane in inactive conformations, and in the ER lumen, ER basically is a store of calcium. So when calcium is dropped into the endoplasmic reticulum, it goes, or stores are depleted. This molecule, which STIM1 or the another isoform STIM2, which is in inactive conformation or in this locked conformation, the EF hand, the calcium sensing domain, loses the calcium and the molecule undergoes massive conformational changes. This leads to the conformational extension of this STIM molecule. And at the extreme C terminus of these STIM molecules, what you see here in blue are these PI45P2 lipid binding domains at the inner leaflet of plasma membrane. So basically, this protein sitting in the ER membrane can form these ER plasma membrane junctions because it directly interacts with this PI45P2. And the membranes come into such close apposition that the distance between them is roughly in this range of 10 to 20 nanometer. And there it makes the life of STIM1 easier to find ORI1 channels. And this functional assembly of STIM1 and ORI1 forms the functional crack channel, where calcium would be influxed from outside of the cell into the cytoplasm. And then from the cytoplasm, the calcium is pumped back into the ER via the action of pump SERCA. And this process is called a store-operated calcimentry. And this is very important for different physiological pathways, and for example, for gene transcription, for T cell activation, for cell mobility, proliferation, and various other functions. And mutations both in STIM molecule as well as in ORI1 have been identified. Both gain of function and loss of function mutations have been linked to immune and muscle disorders, for example. So it's a very important pathway. And there exists three different isoforms, mainly three different isoforms of ORI, ORI1, ORI2, and ORI3. And all three of them are able to individually activate this current, ICRAC, that is the current that is can be recorded through these ORI channels, very calcium selective current. It's conserved in all three isoforms. ORI2, for example, has been suggested to down-regulate the activity of ORI1. And it was shown by different laboratories that when you knock down ORI2, you can augment the activity of ORI1. So basically, ORI2 can form heteromeric channels with ORI1. That's the idea how it would decrease the activity of ORI1. Similarly, ORI3 has been reported to form heteromeric channels with ORI1, and such heteromeric channels have been suggested to be activated by arachidonic acid, for example. So they are uncoupled from stores. And ORI3 has also been shown to confer redox resistance, because ORI1 has a cysteine at the extracellular surface that is oxidized upon oxidative stress. And this, when ORI3 heterooligomerizes with ORI1, because ORI3 lacks this cysteine residue, so this confers resistance to redox. And recently, a recent study from Mohammed Trabak's lab also suggested that ORI channels could form these heteromeric channels, ORI1, ORI2, and ORI3 isoforms. So for ORI1, we quite understand some of the activation mechanisms, but for ORI2 and ORI3, there is no such information. So what I did actually is to use the mutant versions of ORI1 channels. So they are independent of coupling with STIM1, and they are open just by themselves because of a gain of function mutation. So what you see here is a ORI1 cartoon where only two subunits are shown. So the transmembrane 1 domain of the protein, the ORI protein, which has 4 transmembrane domains, the transmembrane domain 1 forms the pore of the channel. So 6 such subunits of ORI channels come together to form the pore of the channel. Just for simplicity, only 2 subunits are shown here. So what was shown here is that if you have in the transmembrane domain 2, if you have this histidine 134 mutated to alanine or cysteine, serine, or threonine residues, the pore of the channel is open. And what you see here are critical residues, the pore lining residues V102 and F99, and it was suggested that this mutation of H134, this removes the bulky side chain of the histidine when you mutate it to such residues, and this somehow allows the rotation of this phenylalanine. So the phenylalanine moves out of the pore, and this gate of the channel is open, thereby the channel can conduct calcium through this. And this is indicative of a communication between transmembrane domain 2 and transmembrane domain 1, that how a change in transmembrane domain 2 can lead to opening of the pore through TM1, so-called TM2, TM1 coupling. So what is key is that this V102, if you mutate it to a cysteine residue, for example, the channel is constitutively open in absence of STIM1. However, the channel remains not selective to calcium. So when you co-express STIM1 with this V102C channel that is otherwise not selective, STIM1 can confer calcium selectivity to this channel. So STIM1 can bring selectivity to the channel by interacting with the channel and changing the pore permeation properties. So similarly, it was found that when you mutate H134 and combine it with V102C mutation, the channel becomes selective. So H134A mutation is basically able to confer calcium selectivity to the V102C channel. On this side, you see the Nexus Mutant. So here, Don Gill's laboratory identified this Nexus region within the ORI1 channel, which when mutated to ANSGA residue, the LVSHK residues mutated to ANSGA residues, the channel becomes constitutively open. And similar to what I said, the V102C gains selectivity when combined with the ANSGA Nexus mutations. And it is believed that this mutation, the LVSHK to ANSGA mutation, causes the flexion of this hinge, which basically then leads to relay of the propagation of this conformational changes, leading to opening of this transmembrane domain 1, the pore of the channel. Also in this study, Don suggested that this leucine 174 in the transmembrane domain 3 forms hydrophobic interactions with leucine 261 in the transmembrane domain 4 exit. As well as it was shown in another study that this phenylalanine at 178 residue position and phenylalanine at 257 position in TM3 and TM4 helices are indulged in hydrophobic interaction mutations, of which disrupt the gating of the ORI1 channel. So overall, this mutation, this Nexus mutant, is highlights the importance of TM4, TM3 coupling in context of opening of the channel. So what you will see in my presentation is using these two ORI1 mutant channels that mimic the STIM1 gated state. I ask the following question, whether if there are any differences in ORI isoforms in propagation of these conformational changes through the transmembrane domains that lead to the opening of the channel. So basically, as you can imagine, I made these mutants in ORI2 and ORI3 isoforms. So what you see here is the, again, the similar structure with two subunits, and the histidine in the TM2 is highlighted here, which is conserved in human ORI2, human ORI3, Xenopus ORI, and Drosophila ORI, and all throughout the evolution, this histidine is a conserved position. So what you can see here is the current through these open channels in STIM1, STIM2 double knockout HEK293 cells. So you see that there is constitutive current with all three open variants of human ORI isoforms, whereas the wild type channels do not conduct any calcium. Yeah, so this is reversed with 0 calcium, and here you see the IV traces of these channels. So what you see here is the NF80 translocation. So when calcium goes into the cytosol, it activates calcineurin, and calcineurin dephosphorylates NF80, which when is present in the cytosol is heavily phosphorylated. And this dephosphorylation of the NF80 by activated calcineurin leads to its translocation to nucleus. So what you see here is wild type ORI1 and NF80 CFP and the overlay. And you see here the NF80 stays mostly cytosolic in the wild type channel, whereas in the H134A open channel mutant, you can see that the NF80 is now translocated to the nucleus. And this NF80 translocation could be observed in most of the cells that expressed these open channel variants of the transmembrane domain 2 of human ORI isoforms. So basically, the take-home message is that this H134A mutation that is indicative of this TM2, TM1 coupling, this leads to constitutive activation of all three human ORI isoforms, suggesting that this TM2, TM1 coupling mechanism is conserved among ORI isoforms. And then we move to the second mutant that I showed you, the Nexus mutant. You can see here the Nexus residues in this hinge region of the transmembrane domain 4 helix. This is the TM4 extension, and here you have the transmembrane domain 4. And this LVSHK residues are conserved in human ORI2, ORI3, Xenopus, and Drosophila ORI, except that in ORI2 and ORI3 you have this arginine and alanine, but previously laboratory of Murli Prakriya had shown that when you mutate these residues, this serine to either arginine or alanine, that does not change at least the interaction of this mutated ORI1 channel with the channel activation domain of STIM1 that causes the activation of the ORI channel. So what you see here, that this Nexus motif is conserved among different ORI isoforms. So you see here the recordings from Flipper where the cells are sitting in 0.2 millimolar calcium, and then they're subjected, or 2 millimolar calcium is administered later on. So you see the control transfected cells, the wild type transfected cells, and the Nexus mutant transfected cells. So you can see a nice increase in the calcium influx compared to the control. Here you see the quantifications, and for quantifications, what you're going to see throughout the talk is this area under the curve of the wild type channel, the area under the curve of the mutant channel divided by the wild type channel. So you get a fold increase in calcium influx compared to the wild type channel always. And this, you see here, similarly, the NF80 translocation assay, that in about 90% of the cells, the Nexus mutant leads to nuclear translocation of the NF80. Then these mutations were made in human ORI2 and human ORI3 isoforms, and surprisingly, human ORI2 ANSCA and human ORI3 ANSCA, these Nexus mutants were not did not exhibit any constitutive activation. And then we further verified it in by electrophysiology, and you see that human ORI1 ANSCA channel, the Nexus mutant, only this was active, and the rest, ORI2 and ORI3, did not show a constitutive ORI current from this channel. So, and then what is interesting, you see that in Xenopus ORI1, the Nexus mutant, where you can note that the LVSHK residues are conserved in Xenopus ORI1, this led to a significant increase in the constitutive, or this showed significant constitutive calcimetry. However, you would see that Drosophila ORI1 or Drosophila ORI, which also has this Nexus conserved, you do not see any constitutive calcium influx through the Drosophila Nexus mutant. So the take home is that the Nexus mutation ANSGA leads to constitutive activation of human ORI1 as well as Xenopus ORI1, but not of human ORI2, human ORI3, or Drosophila ORI. And this leads us to think that why do these differences exist? And so to understand that, we looked into our homology model of human ORI1, and we noted that there is this histidine at 171 residue position in close proximity to this Nexus LVSHK motif. And this histidine, if you would see here in the sequence alignment, that this histidine is actually a tyrosine in human ORI2 and human ORI3, whereas it is conserved in Xenopus ORI1, where we saw constitutive calcium entry, and there is a tyrosine in Drosophila ORI, where we did not see the constitutive activity. So we hypothesized that maybe this histidine at this position, which is a tyrosine in ORI2 and ORI3, is maybe exerting some sort of brake that would not allow the relay of this conformational propagation of the signal that leads to opening of the ORI channels. So we tested that. We made the tyrosine mutation in human ORI1, and we saw that the constitutive calcium influx was significantly decreased by this tyrosine mutation, which was also recapitulated in patch recordings. You'd see here in the dark green traces that there is no constitutive current when this tyrosine is, when this histidine is substituted by this tyrosine residue. And of course, we did the surface expression, because you could argue that, hey, maybe your channel is not on the plasma membrane, but we could rule that out. We see similar expression levels of the Nexus mutant and Nexus plus H171Y mutant on the cell surface. And here you see the NHE1 plasma membrane protein, that similar amounts of cell surface proteins are present on the in the reactions. What we did then is we substituted this this histidine residue to alanine residue or phenylalanine residue. And interestingly, alanine residue did not exert this so-called brake effect on the constitutive influx. Rather, it increased the activity of the channel, whereas the phenylalanine substitution led to a complete block, just like tyrosine mutation. So that's very interesting. And then we did the NF80 translocation experiment. Similarly, you see that the tyrosine mutation, the H171Y, basically decreases the NF80 translocation. So here you see that about 90% of the cells are having nuclear NF80, much higher than the wild type. And you see a nice decrease with the tyrosine mutation as well as with the phenylalanine mutation. However, the alanine mutation does not significantly change the nuclear translocation. So you can see here that in the alanine mutant combined with Nexus, you see a nice translocation of NF80. So the take home is that the H171 tyrosine to phenylalanine, but not the alanine, exerts a brake on the constitutive activity of this channel. Then what we tested is whether other constitutively active ORI1 mutants, known what are indicated here in TM4, in TM2, in TM1, whether they whether tyrosine also exerts a brake on these constitutively active channels. And we see that for the H171Y in the Nexus mutant exerts a decrease on the constitutive activity, whereas there is no considerable decrease in the constitutive activity of other channels that are indicated here, other open channel gain of function mutation channels. What we also did is we tested the A and SHK mutant, which Don also showed that if you mutate the LV-SHK, only the first residues to A and N, it is constitutively active. We could reproduce that. And however, when the tyrosine mutation was inserted in the ANSHK variant of the Nexus mutant, it still led to abrogation of this constitutive calcium flux. So basically, the brake effect of the H171Y is very specific to the Nexus mutant versions and do not affect the other non-constitutively open channels. What we did is we tested the STIM1 dependent wild type ORI1 activation, and we were taken by surprise because, so what you see here is the iCrack, the currents with IP3. So you see the STIM1 dependent activation, and the black is the wild type channel. And the red, what you see is the H171Y in the wild type background without the Nexus mutation. So this is able to activate iCrack similar to the wild type ORI1. This is very surprising. However, the H171Y plus Nexus mutant could not be activated by STIM1. And this was earlier shown by Don that the Nexus mutant has a deficit in binding STIM1. So this was rather expected. However, what is striking that this mutation does not abolish the wild type ORI1 activation. And yeah, so then we try to understand or try to map regions on the ORI channel that could lead to this inhibitory effect of the H171Y. So we asked whether this H171Y, whether this tyrosine needs to talk to the N terminus of the protein or the C terminus of the protein. And this was earlier shown by Don that if you remove the first 74 residues, or if you remove post-Nexus region of the ORI1 channel, the Nexus mutant is still constitutively active. And this is what we see. We see activity in both N-term and C-term deletions, and both were inhibited by H171Y. So the message is that the replaced tyrosine, which exerts this brake effect, does not need the post-Nexus region for the inhibitory effect or the first 74 residue of the human ORI1 to exert this effect. Now, what we did is we looked at these physiological contacts that are important for or that are relevant for the gating, this L174 and the L261 contact and the F178 and F257 contact that I told you before. And of course, the L261 is not L261 in the ANSGA mutant because it's substituted by an alanine. However, in different molecular dynamic simulations and 5 different simulations with the P245L and open tubular aggregate myopathy associated mutant, or the Nexus mutant, or the tyrosine mutant, phenylalanine mutant, or the alanine mutant of H171, we do not see any differences in the contact frequencies between these residues in these simulations about this 174261 contact and the next residue, alanine 175, also did not show any differences in contacts with 261. However, we see very interesting finding from our screen that the alanine 175 is having higher contact frequencies with the tyrosine at 258 residue position here in the transmembrane domain 4. And these contact frequencies are decreased by substitution with tyrosine and phenylalanine. And however, with alanine substitutions, you see comparable contact frequencies with between A175 and Y258. We're still thinking what to do with this information, but this could rather suggest that this tyrosine mutation may be affecting this physiological contacts between TM3 and TM4, thereby not allowing the channel to be constitutively open when tyrosine is present. So to summarize, basically what I showed you is that the H134A corresponding mutation, it confers constitutive activity to ORI2 as well as ORI3. I showed you that the Nexus mutant is functional in ORI1, but not for ORI2 and ORI3. That the tyrosine break mutation does not need the post-nexus or the N-terminal region to confer this abrogatory effect. That it does not, the tyrosine H171Y does not abolish the other constitutively open channels. That it does not block the store-operated activation of the wild type channel. And that there is this A175 and Y258 contact that is higher contact frequency in the Nexus mutant channel and in the alanine mutant channel. So what I will take you through is the very recent data. So just to come back here at this stage, what we did is we then made the tyrosines in human ORI2 and in human ORI3, converted them to histidines, but that was not sufficient to bring constitutive activation of the Nexus mutants of human ORI2 and human ORI3. And that led us to think that maybe there are additional break points that would not allow the constitutive activation because the conformational relay is broken at some checkpoints throughout the length of the protein. So what we did is we took a closer look into the sequence here in human ORI variants, and we see, basically we hypothesized that maybe this tyrosine is not allowing this. If you put a tyrosine here in this position, we hypothesized that maybe this creates a problem in flexion of this hinge and thereby not allowing the channel to function. So what we did is we took the P245 mutation of human ORI2 and human ORI3. So you see that the P245L mutant channel is nicely constitutively active, as well as this F250 that was shown in a screen by Murli, that if you mutate it to a cysteine, you get constitutively open channels. So both these residue mutations lead to constitutive activation of human ORI1. However, when you look at human ORI2 and human ORI3, you see that human ORI2, the corresponding proline mutations lead to constitutive activation. But when you mutate this phenylalanine 211 or 259, the corresponding residues in ORI2 and ORI3, you do not see this constitutive activation of the human ORI2 and human ORI3 channels. So we thought that maybe there are additional break mutations in ORI2 and ORI3. And then what we did is these are the different residues highlighted that are either conserved or not conserved in human ORI2 and ORI3. And what we did is we made a human ORI1 variant that looks like human ORI2 in this part of the transmembrane domain 4, or we made a human ORI1 mutant that looks like human ORI3. So this quadruple mutant versus this quintuple mutant in combination with the ANSGA mutations. And we were surprised to see that human ORI1, the quadruple mutant that looks like ORI2 or the quintuple mutant that looks like ORI3TM4, none of them were constitutively active compared to the wild type channel. So then we asked which one of this is the relevant break mutation. So we checked that this is very interesting, that this phenylalanine is conserved in Xenopus ORI1, which was constitutively active, the Nexus mutant, but the valines are conserved in human ORI2 and ORI3. So we did the F246V mutation in combination of the Nexus mutation, and you see that there is complete abrogation of this constitutive calcium flux of the ORI1 channel mutant. And that was recapitulated in the patch-clamp findings. Then we asked what other residues of these 6 positions that I showed you could be relevant, and we went ahead to the mutation. We didn't mutate the 249 because this was a valine as well as in Xenopus ORI1, which we know that was constitutively active. Then we moved to 251, which is a valine in O2 and O3 that did not create any problem in the constitutive activity. We moved to this V252A, that's ORI3-like. We saw a small decrease in the constitutive activity, but not a complete block. So what we did is then we moved to the following residues. We made human ORI2, these 2 residues look like human ORI2 in human ORI1, and we see that the constitutive activity was rather increased, but there was no inhibitory effect. However, to our surprise, when we did this ORI3, like this triple mutation following the F246, there was a complete block of the constitutive activity. So V242A, which I showed you that there is this minor decrease when combined with these two additional mutants, led to a complete abrogation of the constitutive influx through the Nexus mutant. We did the same thing then with F246V. We deleted the post-nexus region, and this would still block the constitutive influx of the channel. So the C terminus is not relevant for F246V to block the constitutive activation. We did the same thing. We combined with different constitutively known mutants. However, this time, very interestingly, there was a significant effect of the F246V mutation on the V102C, H134A, and P245L, all indicated here. Strikingly, the F250C mutant that I showed you was not active for ORI2 and ORI3 variants. When it is combined with F246V to look at like ORI2 or ORI3, we see a complete block of this constitutive activity for this double mutant channel. And this brings me to the summary of the second part, where I showed you that this F246V mutation in TM4, which is the relevant residue for ORI2 and ORI3, this inhibits the constitutive activity of the Nexus mutant. Showed you that it also inhibits the F250C constitutive activity. I showed you that it doesn't need the post-Nexus region to exert this effect. I showed you that it does only a small inhibitory effect on other open channel mutants. And I showed you that when you combine it with other residues, which are ORI3-like, it leads to a complete inhibition of the ANSCA nexus activity. So to sum up, all these experiments suggest that the reason why, first of all, it is indicative of that human ORI2 and human ORI3, the way the conformational change is propagated through the channel may be different for human ORI2 and human ORI3. And next, that these residues that we are talking about, the H171 or the F246, these residues could be exerting sort of a break effect into this relay, thereby might translate to different activation mechanisms of the ORI channels. And last, we substituted both of these in combination, and it was still not sufficient to rescue the constitutive activation of O2 and O3 nexus mutants, which might tell that there might be additional breakpoints into the human ORI2 and ORI3. And that brings me to my acknowledgement slide. I would like to thank Matthias, who gave me a chance to do all this work in his laboratory, Kristen Peinelt, with whom we did the electrophysiology recordings, and Bartek, who did the patch map recordings, and Jan, the master student with whom I did the experiments, Gerke, who did the MD simulations, Irene, who did the NF80 translocation experiments, Giuseppe, who did the biosimulation experiments, and Gabriel, who worked with me, recently started working with me as a master student. And then, of course, I would like to thank all the other Hediger, Peinelt, and Frisch-Auf lab members, which contributed to this enormous amount of data. And I'm open for questions, and very sorry for the 3-minute delay.
Thank you, Rajesh, for that really interesting talk. There are already several questions in the chat box, so I will start with the first one from Antonio. And he's asking when H134 is mutated to serine or threonine, could these residues be phosphorylated? And if yes, could this phosphorylation block calcium influx?
Well, first of all, these are all active. The H134 mutant variant here, right? So these mutants were, they were active actually, the open channel mutants. So I would say that this is not phosphorylation dependent effect that we are seeing the constitutive activation, because you see that other residues that are not phosphorylated also lead to this constitutive activation. What this phosphorylation could do on the serine threonine residues, it could bring in additional regulatory levels, we don't know.
Antonio, is your question answered by that? Is he still listening? Jonathan, your question is still active?
My question is answered.
Your question is answered, good. Shmuel is asking, are the ANSCA, ORA2 and ORA3 mutants activated by the STIM and SOR?
That's, I think Shmuel asked, suggested this experiment, but we still haven't managed to test that, whether SOAR would activate human O2 Nexus or human O3 Nexus mutants. We know that human O3 Nexus mutant responds to 2-ABB at least, but no, we have not yet tried with SOAR or STIM. Yeah.
Shmuel?
But I think it would be an interesting experiment to do.
Yeah. Shmuel, is your question answered?
Yeah, absolutely. But still, thanks for great talks anyway.
Thank you, Shmuel.
Well, really great data, Rajesh. I also have a question. When you checked for these A175 Y258 contacts,
just let me get to that slide.
Yeah.
Yeah, here. Right, yeah. This tyrosine is just preceding to, just next residue after this F257. And A175, Don had showed in the past that if you mutate to a lysine residue, you disrupt this physiological coupling, basically.
Yeah, exactly. Because I didn't get the slide, it was a bit quick, but actually, you also see here with the H171A mutant that it's not, there is no effect. So it's
right. So we could try, for example, a cross-linking experiment to see whether this contact is relevant or whether it would affect the activity of the Nexus channel, for example.
Yeah, great.
An interesting experiment.
Are there any more questions from the audience? We can also go like to an informal discussion where we can see each other also and talk to each other if you want to.
That was super interesting, Rajesh. Thanks very much. I'm happy to, as I really directed, if people want to, if we end the formal part now and we can stop the recording, and